when people say Johnny's just that way, he's wired differently.
Mm-hmm.
That's actually pseudoscience right now because we've never been able to see the brain's wiring clearly enough, or really at all, to know whether that's the case. It's just a supposition. But with the technologies for revealing and mapping the connectome, we can start to address those questions.
I'm Insoo Hyun, Director of the Center for Life Sciences at the Museum of Science. Today, my guest is Dr. Sebastian Seung, who's Professor of Neuroscience and Computer Science at Princeton University. We talk about the brain's connectome. What is it, and how does it make us uniquely who we are? So, Sebastian, it's so great to have you here. Thanks for joining us.
Fantastic to be here.
So I did a little bit of background research on you, and I found something out really interesting. You and I have something very interesting in common. So your father left North Korea, I believe, during the Korean War when he was a teenager.
That's
And
right.
my father left North Korea when he was 10 years old.
Oh,
in
wow.
The Korean War.
So we might like the same foods.
Yeah, I'm sure we do. The Korean naengmyeon from North Korea. I'm a big fan. So your father left North Korea. He eventually came to the U.S., and he was a philosophy professor.
That's right. That's right.
And this is in UT Austin?
Yes.
Now, is that where you grew up? You grew up in Texas?
I grew up in Austin, Texas. I was born in New York City, but I grew up in Austin. And my father was a philosopher. He was a battlefield convert to Christianity, and that religious questions are what drove him into philosophy.
Interesting. Right. And do you think that had some effect on some of your earlier interests? Was philosophy something that you kind of heard about or ever thought about when you were younger?
Well, my mother is a musician, and my father said, no son of mine will do the humanities. And my mother said, no son of mine will do the arts.
Uh-oh.
So I guess that left science. But in the end, I became a neuroscientist. So I guess I ended up in the area of science that's closest to philosophy. Perhaps that's my misfortune.
Well, that's interesting you say that because so much of your work, I think, has a really interesting adjacency to philosophy. I see that your work itself, in an indirect way, raises those questions as well.
Certainly it does. I guess my father used to say is, what he used to say was that philosophy is whatever hasn't been successful in science yet. And so I would say my work, though, is about trying to make neuroscience into real science.
Mm-hmm.
In some ways, the field is pre-scientific. That's why it's close to philosophy. But it's a struggle, right? That studying the brain is very hard. It's hard to make it into something, a science like physics. So you can't really escape the philosophical aspects of it.
So I guess unlike your parents, my Korean parents encouraged me to go into philosophy.
Oh, really?
I got a PhD in philosophy. And so now I work in bioethics, which is, again, adjacent to science, but from the philosophical side. But when you were younger, though, I think you were initially interested not just in science, but in physics, right?
Yeah, specifically in physics.
In physics. What was it about physics? And then why did you get out of physics? What drew you to physics? And then why did you leave it?
Well, physics, of course, you could argue is the most fundamental of sciences, right? The idea of finding the basic building blocks of matter out of which everything is composed. It sounds such a cosmic kind of quest. I think physics is really very captivating. Also, it's mathematical. I loved mathematics. But I guess when I finished grad school, by that time I was growing somehow dissatisfied with physics because it seemed like it wasn't enough to divide everything into the basic building blocks, the fundamental particles.
Mm-hmm.
What we wanted to understand was the problem of emergence. When you have collections of these elementary particles, you can get something very different, very different kinds of behavior that don't seem to be explained so easily by the properties of the parts, right? So the classic example in physics is diamond and graphite. They're both made of the same carbon atoms, but one of them is soft and dark, and the other is shiny and hard. They're made of the same atoms. You can't just give you can't give a graphite ring to your fiance and say it's just made of the same atoms, right? They have very different properties.
It's basically a diamond.
It's basically a diamond.
Look at it the right way.
Just rearrange the atoms in a different way. But one of the greatest triumphs of 20th century physics is understanding why the different organization of atoms in these two kinds of matter lead to very different material properties. But once you understand diamond and graphite, then you might say, hey, I'm not satisfied with that. I want to understand other kinds of matter that seem harder to
Mhm.
understand. Like, what about living matter,
Mhm.
right? What makes a living thing, it's the same atoms, what makes a living thing alive? And of course, the ultimate challenge is what makes an intelligent
Mhm.
thing intelligent or even conscious? How do you explain that if it's just the same atoms?
Yeah, I want to get into the idea of biological emergence in a moment, but let me just ask you. So the switch from physics to neuroscience, so that was in grad school. That was,
Well,
or
at the
did that
tail
start in
at the
college?
tail end of graduate school, my PhD advisor, David Nelson, who's a wonderful, incredible professor of physics at Harvard University, he sent me off for a summer job at Bell Laboratories.
Mhm.
And that's where I first encountered physicists who were thinking about brains and artificial intelligence
Oh,
and so on.
wow.
And so that's how I got exposed to these ideas.
You entered Harvard University at 16 years old.
That's right.
Looking
That's right.
back, would you have done that again? Like, would you have waited a longer, or how was that entering that university at age 16?
Well, on the one hand, it is strange to be a 16-year-old. You're really immature.
Hmm.
You're in college. But on the other hand, I found a lot of people who were similar to me, right?
Hmm.
Real nerdy people who they didn't fit in well in high school either.
Hmm.
And so it was perfectly fine to be at Harvard as a 16-year-old. And in fact, quite a few students in those days were younger than average. There were 7-year-olds, 16-year-olds, and even there was this genius classmate of mine who was 15 years old.
Wow. So it already is an island of misfit toys, so why not?
Yeah,
Yeah.
it's and maybe I would have enjoyed college more.
Well,
Uh,
you couldn't drink even when you graduated, right? You graduated at
right.
20?
But I would have enjoyed college more maybe if I'd been older, but certainly going to college was better than staying in high school,
Yeah.
which I didn't enjoy it to the same degree as college.
Let's talk about the connectome because that's really where people most associate your achievements and your work, and that's what you've been really focusing on. So tell us how would you explain what the connectome is to just a basic lay audience?
Well, a brain is a network of neurons, right? And every network, you can map it out. So think of the
Mm-hmm.
map in the back pages of airline magazines,
Hmm.
right? There's a bunch of cities and there's flights between cities.
Hmm.
So in this same way, if we just take every city and replace it by a neuron and every flight between cities by a synaptic connection between neurons, we could think of a map. But that map for your brain is huge, right? It's 100 billion cities and thousands of flights per city. So it would take a really thick airline magazine to contain that map. But
Hmm.
that's the map that is really the goal. Attaining that map is the goal of connectomics, at least the technological
Yeah.
side.
So is the connectome for every person? Does it basically look the same, or would you say that they look radically different? These maps.
Well, nobody knows for sure because we don't have a
You don't have that,
connectome
right?
for a human, right? Um, the only connectomes that we have are for small brains or small nervous systems like worms and more
Mhm.
recently
Mhm.
a fruit fly,
Mhm.
right? And what we do know is that in those animals, for example, in the fruit fly, the connectome is very similar from fruit fly to fruit fly across
Mm.
individuals,
Okay.
right? And that is consistent with our intuition that um, a fly or an insect is kind of like a wondrous machine, right? It's like a mass-produced object. But there are differences. There are differences between normal individual flies. It's not exactly the
Mhm.
same. And some of those differences are likely associated with the limited ability of flies to learn from experience.
Mhm.
Now, in humans or in animals with larger brains, um, there's much less literal reproducibility as far as we know between one human brain and another. For example, we're guessing that the number of neurons could vary greatly because we know that the size, the volume of human brains varies quite a bit, right?
Mhm.
But some things can be found over and over again. So types of neurons, maybe not individual neurons, but certain types of neurons, they can be found probably in every normal human being.
Getting back to the metaphor of the connectome being a map, have you mapped out just one fruit fly's brain, or have you done many? Because it took so long to get the first one, right? So,
Right,
yeah.
right. So I was part of one effort which mapped out one female fruit fly brain.
Mhm.
And recently, another team, they've released just a few days ago, they released the map of a male fly brain.
Mm.
And there are differences.
Well, like what?
There are certain types of neuron that are male specific. They're found in the male brain, but not in female brains, and vice versa.
That's interesting. Do people try to infer from that there's like some observable or kind of like, that matters in some way?
Oh, yes. Some of those male specific or female specific neurons are known to be important for sex specific behaviors.
Okay.
For example, if a male fly is courting a female fly, they both have behaviors that are sex specific.
Mhm.
And some of those neurons have already been shown to be related to that.
Interesting.
It's true in this worm C. elegans.
Mhm.
The male nervous system has some number of neurons that don't exist in the female nervous system, and many of them are involved in mating.
How long did it take to map out the first fruit fly's brain?
From start to finish, from the time that there was one researcher who sliced the fly brain into 7,000 slices, and from that to the imaging of the brain to the actual image analysis and annotation and so on, about 10 years elapsed.
People are working on the mouse brain right now, right? That's like
Sure,
the next.
sure.
Right.
Well, they're working on all kinds of brains,
Okay.
so small brains. There's many efforts going on to map mosquito brains or fish brains and so on and so forth. But scaling up to a mouse brain is a lot harder because that's maybe 10,000 times larger
Hmm.
than a fruit fly brain, over 10,000 times
Yeah.
larger. So that's 10,000 times more data to handle.
So do you think the human brain would be mapped out in your lifetime, or they have no idea?
Well, if I eat right, take lots of Geritol, well, it depends. It
Yeah.
depends on both on the march of technological progress. So we all know that although, 10 million times more data might be involved, we can imagine that computers could be 10 million times more powerful in a few decades,
Mhm.
maybe less than a few decades, right?
Yeah, yeah.
So it's within the realm of possibility,
I see.
but it also depends on, do we care enough,
Mhm.
right? So there's two questions. One is it technologically feasible? And the other is,
Mhm.
how much do we care?
Do you think people should care? And why should we care to do this?
Well, there are a lot of potential applications. Obviously, I can talk about the problem of brain disorders
Mhm.
and mental illness and so on. There's a lot of reasons, practical reasons, to want to know more about our brains and know ourselves, right? But there's also just, it's just something fundamental, right? It's going to be, if you think about it, the first time that we can map a human brain, that's just a fundamental step forward in the history of humanity, right? So it's an adventure, and it's like a quest. It's a quest to know ourselves, and there's bound to be practical applications, but there's something bigger than that.
When I talk to scientists, especially people who are trying to find more public funding, this involves a lot of science communication with funders, with the public. It's really interesting that it could be more easily made. The case could be more easily made if you say, oh, this research could lead to therapies for patients with this disorder. Like there's some like clinical translational benefit. And that's like an easier sell, I think, to the public. Harder, but I think just as important for discovery is just what we call basic or foundational research, where you just want to know, how does something work? And it may have, as you were saying, it may have clinical benefit down the road. It may have a practical application, but that's not what's driving the researchers at the current time. So it sounds to me like you're more in that latter category.
Well, I think they go together, right? Just fundamental advances in our understanding, if they're really fundamental, are just bound to change the world in practical ways too. That's the first thing I'd say.
Mhm.
And the second thing I would say is that the public is also cares about basic research, right? There's a if we send astronauts to Mars, whether or not there's any practical application, I think the public is excited by that,
Yeah.
right? It's exploration. It's part of what humanity does, right? We want to explore, we want to know the unknown, and everybody gets that at some level, I think.
I used to be the vice chair of an IRB at a medical school. And when we train new IRB members, I know this is not human subjects research, not yet what you're talking about. But when we train them, what we train them is, and this is in the guidelines in the training manuals from the federal government, as an IRB member, you're not supposed to weigh in your deliberations, take into account how discovery in the protocol, how the discovery in that research track might affect how people think about themselves, like in a broad sense. Which is I thought like kind of a fascinating kind of thing. I like to say that's not really in the risk benefit calculus. But obviously, many people are concerned about how sometimes science could change how we perceive ourselves. Maybe not in always the best ways. Some people might say, the Human Genome Project basically forced people or encouraged people to kind of reduce themselves all the way down to like they're just their genes. Do you think on your road to kind of basic fundamental discovery, whether it's in the fruit fly brain, rat, mouse brain, or human, this type of research, looking at the connectome, trying to understand that better, do you think there's a possibility, this kind of gets a little bit more philosophical, how that might maybe change how people think about themselves and about what it means to be a person and identity? Do you think there's that possibility? I'm not saying it's a bad thing, or it's just something that, like it's a risk that we have to accept. But do you think that it leads to that? It leads to that kind of like reorientation of how we view the human mind?
Well, I think that science, of course, always has implications for people's worldviews, but it doesn't really dictate one worldview or another. I mean, I think in some sense, maybe it would dictate the idea of rationalism or empiricism, that if we look at the data, we'll learn something.
Mhm.
If we reason about it and don't just take authority word for it, then we make progress. I think maybe those values, of course, are part of the kind of scientific uh, worldview or scientific way of doing things. But the actual discoveries of science, they could be interpreted in many different ways.
Mm.
Science can make people less religious. It can make people more religious. I think there's something that goes beyond science in affecting how people view the world, right? It's
Mhm.
just one ingredient.
Yeah, years ago, you gave a really popular TED talk and you posed a question, am I my connectome? Do you still frame it in that way? Like, do you still think that there's a connection between the connectome and understanding it and personal identity? Like what makes us who we are?
Oh yeah, that's really right. So the slogan was "You are more than your genes. You are your connectome."
Mhm.
And the reason, I mean, that really was to express an idea in some way, a hypothesis, right? That whenever you have an experience, your connectome can change,
Mhm.
right? So neuroscientists believe that changes in synaptic connections are the most fundamental aspect of plasticity and the way in which you are affected or shaped by your experiences, right? But at the same time, the connectome is shaped by genes.
Yeah.
So it's really where nature and nurture meet, right? That's really, to me, what makes it really fascinating, right? We've had, centuries or millennia of debates about nature and nurture. But the connectome is this entity where these two forces are meeting. And we can start to study that. We can start to study that. And I mentioned, male and female connectomes of flies, right?
Mm.
We already know that these differences in neuron types and their connections are related to the different behaviors of male and female. And so it's not a far stretch to start thinking about, and we don't know, of course, that's flies. That's not the same thing as humans, right? But it's not a far stretch to open up all the other questions of identity, right? What causes the differences between normal human individuals? when people say Johnny's just that way, he's wired differently.
Mhm.
That's actually pseudoscience right now because we've never been able to see the brain's wiring clearly enough, or really at all, to know whether that's the case. It's just a supposition. But with the technologies for revealing and mapping the connectome, we can start to address those questions.
You once used the metaphor of a riverbed. Could you unpack that for us a little bit? What do you mean by that?
The connectome is about this. The connectome is really a description of the structure of a brain, right? I said that it changes, but it is material structure, and so it presumably changes relatively rapidly. I mean, sorry, relatively slowly, right? Imagine a forest, right? Imagine there's a forest, and you go back year after year. It's different, but the trees are basically the same. The main branches are basically the same. The leaves have changed and so on. And so an adult brain is presumably like that. And even in the developing stage, it's going to be growing slowly. So that conflicts with other people's, another notion of personal identity, which is my conscious self, right? Which just changes all the time. Right now, I'm happy. The next moment, I'm sad, I'm hungry, I'm irritated and whatever. That's changing from moment to moment. So how could that identity be related to this idea of identity in the connectome? But at the same time, husbands and wives ask, will he ever change? Will she ever change? Right? So we know that something is stable about people too. And so the idea in neuroscience is that there's the activity of neurons. That changes from moment to moment, but then there's the connections, which are really much more stable. And so one analogy is the river and the riverbed, right? The river, the water is constantly moving, but the riverbed changes only very slowly. And it's a kind of two-way causation that the riverbed tells the water how to flow, but the water shapes the riverbed.
Mm-hmm.
And it's the same idea, these two kinds of identities. So the connections determine which activity patterns can be expressed by your brain. That's why you're not a totally different person from moment to moment, right? But at the same time, the activity, when you have an experience, the activity can leave an impression on the connectome.
Wow.
And so that's the water shaping the course of the riverbed.
That's a great metaphor. And by the way, it kind of recalls a little bit of Bruce Lee. Be like water. I was thinking that too. I was just thinking talk about the water.
Be like water.
Well, now I want to get back to philosophy and science because there's some interesting, really interesting, like parallel conversations I think happening in the philosophical scholarly community it has been for centuries, and kind of the ideas you're bringing forth. So there's been a pretty prominent, but now not so popular theory of personal identity. What makes the person the same person over time? Like what makes you the same person that I met, years ago? And John Locke, the philosopher, thought it was a memory criterion. It's like the continuity of memory is really because, bodies change and could change pretty radically. But as long as there's that continuity of memory, then it's the same person. I've always been a little bit kind of skeptical of that explanation because to me, it kind of presupposes personal identity to explain it because you can have a memory from someone else. You can only have your own memories. So that idea of like memory continuity kind of presupposes there's this thing that's continuous throughout, and you're kind of presupposing personal identity. So I wasn't always super happy with it. But what are your thoughts about kind of like this? Are they at least in the right ballpark with that theory? Are they getting to the right direction? Is it memory continuity? Because there's got to be, well, obviously, then we know there's connectome that's pretty stable over time. Of course, things can change pretty radically with head injury. And there's very interesting questions of is that the same person after the massive head injury? What are your thoughts about the memory criterion for personal identity and kind of what how you think about the brain?
Memory is it's a hard word because how do you even define what a memory is, right? So there's the memory of what I had for lunch. There's the memory of who's the president, the third president of the United States. There's the memory of my tennis swing and so on. And then there's just things that people do, maybe habits they have, right? Or mannerisms they have.
Mm-hmm.
Are those memories?
Mm-hmm.
And maybe they're stable over time, but it's so I don't know exactly what Locke meant by memory.
Yeah. There was a really famous critique of the memory criterion for identity. It's kind of a brilliant one. So identity, if A equals B, then that's what we mean by identity, is that equal sign. So if person A is equal to person B and person B is equal to person C, then by transitivity, A is equal to C. And so here is a counterexample. So you are interviewing an old general throughout his career. You're the biographer. And in your notes, you say, "Old general, can you tell me about an experience of when you were a young officer?" Because, I remember when I was a young officer, I stole the flag from the opposition. I waved that around and it inspired everybody. And so that's interesting because I'm flipping back. Apparently, the interviewer is very old. Flipping back at my notes, I asked the young general, "Tell me a memory about your childhood." And that person B, said, "I remember stealing apples from an orchard when I was a child." And the old general says, "Well, I don't remember that." So then you're not the same as, A.
Right.
It's really interesting. So we know memory is kind of it. You said, "Well, what do we mean by memory?" But it's not like it goes. You have necessarily memory connections from the beginning all the way to the end of your life. There are disruptions that happen,
Sure.
even without brain injuries. So that's a really interesting.
Also, we have false memories.
There's that problem as
Right.
well, right? And actually, it's quite easy apparently to get people to think they have real memories when they're
Right.
false, right? So
Right.
there's this whole reliability issue, and you can only kind of through introspection kind of know whether or not you are the same person over time if that's true. So there's that problem. There's a more kind of current view. Derek Parfit has a view that it's actually there's no one self that you are throughout all your life, but you have different stages of selves. So like there's no one England that medieval England was, and industrial England was, and that modern England is now. There's no one England. You have different stages of the thing, or like a basketball team. You switch players in and out. It's still the Celtics, but it's not really the Celtics of all time. And so human identity is kind of like that. So do you think that's more on the right track based on what you study and what you know? Or does that accord with what you're doing?
Well, I don't know what to say about this because clearly there is, in the case of a brain, right, there really is a material object that we can trace its path through space-time. And there's a single material object that goes along, and we could say that whatever that brain does is somehow an expression of that self, right? Now, the big problem would come if we could somehow extract the information from that brain and make another, make a copy, right? We can do that with laptops, right? We have a laptop, we just copy, the data from one laptop to another laptop, and now we have a second copy of it. So I guess I don't really, that seems to be a much bigger challenge to the identity theory, right? Because that, if brains were like laptops and we could create another brain which had the same memories, then what does that mean to identity?
Let's just do the following thought
Yeah.
experiment. Years in the future, we have the connectome for the
Right.
human brain. And we can kind of, maybe it's easy to do, it's quick to do for everybody. And someone proposes that, as they near death, they would like to sort of transfer that, because you're talking about computers, right?
Right.
Transfer that connectome somehow to in some other way.
Right.
And that they will then, live on. And so if that's true, then maybe the memories carry on. And so whether you believe in the memory criterion or the successive selves thing, those are two pretty close successive selves, but it can branch off. The successive self idea is also very interesting because you can have multiple versions that kind of branching off at the same time. You seem to kind of want to start by saying there's some intimate connection between who we are individually and our connectomes.
Right.
But then the connectome, if you do this thought experiment under the right circumstances with the right technology, could be sort of transferable in some ways.
Well, that's the ultimate test. The ultimate test of the slogan, "You are your connectome," would be, can we use that map of connections to build a simulation of you, right? And of course, nobody knows the answer to that question. But what I can say is that this science fiction idea of mind uploading is now becoming mainstream science for flies, right? So we can imagine, and this is a kind of experiment, a kind of thing that people are trying to do. We take a fly connectome, and we try to simulate its brain, We hook it up to a
hmm
virtual body, and then we see how it behaves, right?
Yeah, yeah.
And let's say this fly was a fly that happened to usually turn left. Such flies,
hmm
we have a fly. Oh, it likes to turn left. Oh, this other fly likes to turn right. If that's all determined by the connectome, then we would hope that the simulation of one turns left and the simulation of the other one turns right. Now that hasn't been tested yet. Or if we could find some flies seem smarter than other flies.
Really?
Okay.
Is that like a pretty big,
So,
pretty big spectrum?
you make them do tasks. They seem a little smarter than others. Could that come out of a simulation, right?
Mhm.
So certainly certain mutant flies are dumber,
Okay.
right? So at this question of identity, of personal identity, it's kind of weird, but you could explore it already now with animals with simple
That's
brains.
so interesting.
Start at the beginning stages. I'm not saying
Right,
it's solved yet,
Right.
right? I referred already to male-female differences, right?
Yeah.
So can we simulate a male brain and simulate a female brain and see how the different behaviors emerge from the simulation? That's a challenge for science now. It's not science fiction.
So as this work progresses, what really excites you the most about the field?
There's many different feelings that I get, right? So of course, science is about trying to discover the objective truth and so on, but scientists are like other humans. They're driven by all kinds of feelings, right? So hunches, um, controversies, the desire to prove that other people are wrong and they're right, all those things. And so, one of the aspects of the connectome that's very interesting historically is that um, many neuroscientists thought it was dumb to want a connectome. They thought that this data would be, they say, fine, you'll get all this data. It's going to be useless,
Hmm.
right? So it was a technological struggle to get a fly connectome, but it was also a kind of political, cultural struggle because many neuroscientists, actually the vast majority of neuroscientists, thought it was not interesting.
Do you think they think it's interesting now?
Well, the fly researchers have all embraced the connectome. They all use it. But let's say you're somebody who studies humans or somebody who studies monkeys or mice, more complicated brains. Many of them might still say, yeah, that's fine for flies. You can study your piddling flies. That's nothing like a real brain, right? So the controversy, I think, goes on, right? It's going to go on for some time.
But in defense of the fly, though, I mean, the fly brain has some remarkable similarities to the human brain, doesn't it? I mean, aren't there some aspects of it? I mean, that's why you study the fly brain to some respect.
So yeah, just to step back, there are similarities between flies and humans. It seems incredible, but for example, the genomes, right? You can find genes in the fly genome that are homologous to the human genome. In fact, I forget some kind of factoid, 60% of the genes, you can actually match them. They're not exactly the same sequence, but you can match them, right? Now, everybody's accepted that genetic similarity, but fly brain and a human brain, they do look very different. Obviously, there's the size, but it is true that there are some principles of the way the circuits are organized that are shockingly similar. So, for example, the olfactory system, the neurons that process smells, right? They're not the same neurons in flies and humans, but some of their principles of connectivity are very similar to each other. And the same can be found in the visual system. So these are probably parallel evolution. Sometimes you can, in a mouse and a human, if a mouse has a kidney and a human has a kidney, well, there was some ancestor that both had the same kidney, right? But in the case of a fly and a human brain, if we find similarities, maybe that's not parallel evolution, that's convergent evolution. To solve the problem of smell, to solve the problem of vision, some similar solutions were found by evolution.
Now, just to get an idea of how complicated a human connectome must be, if you took all the human neurons and lined them up in the brain and all the connections, like how long is that?
Right. So the statistics, right? 100 billion neurons, uh, many thousands of synapses per neuron. People like those factoids, but the one that I like the best is the length of the wiring in a human brain, right? So the wires of the brain, in scare quotes, we say wires. Those are these long, slender branches of neurons. If you laid them all end to end, our best estimate would be that would stretch millions of miles, right? So that's multiple round trips
Around
to the
the,
moon,
yeah.
multiple round trips to the moon. And so that's crazy, right? That's all packed inside a pretty compact volume, right? Now, a fruit fly is a really small brain, tiny, right? A little sliver, a little smidgen. But still, our study found that it's more than a football field's worth of wiring, packed inside this tiny brain.
Wow, it's remarkable. So while
And it's a mess. Well, it's a mess. It's all tangled up together. It's very organized though in a fly brain. Fly brains look a lot more organized than our brains.
It's good to know.
They're little machines.
So while I have you, I want to ask you about two things that are kind of like pretty hot in the area of bioethics that I work in. So one is human brain organoids, and human brain organoids are just a few million cells as compared to what you're saying the actual human brain is. But it's kind of along the lines of maybe what you might find in a fly brain. And what people are worried about with the human brain organoids, which are these little models grown in a dish that kind of recapitulate different regions of the brain, is that you might have emergent, like human-like consciousness. Now, walk us away from that cliff. Like what from what you know about the connectome, because it seems like it's all about the connections, the wiring. You wouldn't expect just like a clump of cells, even if they're self-organizing into like what looks like brain tissue, a region, that you wouldn't expect there to be any kind of like experience. Or would you say that it's not clear at this point, even from what you know?
I don't know that much about organoids, but like you said, it's very early stage. You can grow cells that are like neurons in vitro. Whether they are organized into brain-like structures, that's a kind of a judgment call. But it's clear that the goal of the field is to make those structures more and more like the ones that are in real brains, right?
Yeah.
So that may be the sort of slippery slope along which you as a bioethicist would get worried. But still, there's other experiments, right? There's experiments like the fellow at Yale who is keeping
That was going to be
brains.
my next question. Is
Right.
the brain X group? So that they keep outside of the body
Right.
human brains that are perfused and are metabolically
Oh, I didn't know about
active.
the human brains. I only knew about the animal brains.
No, they have
Yeah.
human now.
Okay.
They do human, and so they're able to indefinitely keep them metabolically active.
Right.
And
Right.
would that be a system that would be interesting to you to study?
Well, I think that, of course, raises a much thornier set of ethical issues, right? So then you have a full-blown brain. You don't just have cells that are drawn from some particular part of the brain, but you have a full-blown brain with all the structure and so on. And so if those neurons are active, then one could worry about the brain in the vat situation. So yeah, I think that's something that you would definitely want to worry about if you're a bioethicist.
So as you see this field progressing from fly to mouse to who knows what else after, maybe eventually to human, where do you hope this work will lead? Like, what are your aspirations for this type of work?
Right. People often ask this question, but in some sense, it's a hard one to answer because my expectation is that the connectome will simply transform every question in neuroscience, right? If you think about the way that the genome unfolded, right? It's just taken for granted. Everybody who studies biology, who studies medicine and so on, they use genomics as a fundamental technology and they take it for granted. And so in the same way, we've struggled very hard to develop the technologies of connectomics. And a lot of what I do these days is trying to make that technology work, be, cheaper and faster and so on, so that the maximum number of people, researchers can use it to understand how the brain works. So there's so many interesting questions. I can't answer them all. And in fact, the most interesting questions will be probably ones that I never anticipated, right? But I will give a few questions that certainly drove us in the beginning. One of them is the issue of miswiring, right? So there are, broadly speaking, two kinds of brain diseases. So one kind is Alzheimer's and Parkinson's, where brain cells die, right? That's kind of we don't know why they're dying. We don't know how to prevent the cells from dying, but at least we know that something is clearly going wrong in the brain. That's the first clue, right? But there's plenty of brain disorders. If you look at the brains, they look normal. Like autism, schizophrenia. The neurons are totally healthy. They're not dying.
Mm-hmm.
So what could be different? What could be different? And since the 19th century, already scientists speculated that the neurons are perfectly healthy in schizophrenia. They're just wired up in an abnormal pattern, right?
Mm-hmm.
And so that's been a really deep hypothesis that's been completely untestable.
Mm-hmm.
But now, with the technologies of the connectome, we could start to trace all that wiring, map out those connections, and really see if there are miswirings underlying these brain disorders, which we call brain disorders, and yet we can't actually see anything wrong with these neurons, right? So, imagine you go behind a supercomputer and you find a tangled up miles of wiring, right? How would you know if there was a wire in the wrong place, right? It's really, really difficult to do that. And think about the million miles of wiring in a human brain. How can we know whether Johnny's just that way, he's wired differently? It's just not known. So that's a huge challenge, and figuring that out would be tremendously important. Obviously for all the dollars and the emotional pain of mental illness, but also just understanding something really deep about ourselves.
Well, thank you so much for your insights, and I really enjoyed talking about the fly brain with you and implications for the human brain and our identities. Thank you so much for being with us.
Thanks a lot. It was fun.